The overall goal of the following experiment is to prepare viable Drosophila embryos with a permeable eggshell to test the bioactivity of introduced small molecule drugs or toxins. This is accomplished by first treating developmentally staged embryos with dilute bleach to remove the outer chorionic eggshell layers. The second step is to permeabilize the embryos with a solvent that depletes the thin waxy eggshell layer that blocks small molecule solutes. The third step is to treat embryos with a drug or toxin and a dye to simultaneously gauge the permeability of the eggshell and introduce a compound of interest. After further development, the embryos can be either directly imaged or processed for immunohistochemistry. Ultimately, the results can show the activity of a drug or toxin on the development of tissues as determined by fluorescent microscopy with immunoreagents or vital dyes.
Today I will be presenting our method of permeabilization of Drosophila embryos for assays of small molecule activity. The main advantages of this technique over existing methods, like the use of conventional organic solvents such as heptane to permeabilize the eggshell, is that our embryo permeabilization solvent, or EPS, is water miscible and relatively non-toxic. These properties facilitate embryo treatments whereby solvent exposures can be more precisely controlled and can be carried out in physiologically relevant media, both of which result in greater embryo viability. I first had the idea for this method when I contemplated how to remove the waxy layer of the eggshell. Being a skier, I reflected on the common use of citrus solvent to remove the residual wax from the base of skis. This prompted me to